Hello everyone, my name is Yingnan Zhang. I come from Ohio University. Today my topic is multiplex-GAM, genome-wide identification of chromatin context yields insights overlooked by Hi-C. Genome architecture mapping, which is called GAM, is a technique that can identify chromatin interactions. The principle behind GAM is that DNA loci that are physically close to each other in the nuclear space are present in the same nuclear profiles more frequently than loci that are removed from another. In the original GAM experiment, thin slices from individual nuclei are isolated by cryosection and laser microdissection before the DNA content of each slice is determined by next-generation sequencing. The multiplex-GAM is an improved version of GAM. In this variant of GAM, multiple independent nuclear profiles can be added into a single tube and then sequenced together. It can cut down on both labor and reagent costs. To test this idea, we used a dataset of 481 single nuclear profiles sequenced individually, simulated multiplex-GAM sequencing of 2 or 3 nuclear profiles. We combined 480 of the single nuclear profile dataset and generate 240 or 160 in silico GAM samples containing 2 or 3 nuclear profiles. We then recalculate co-segregation matrix from this simulated multiplex-GAM datasets and found that these were visually highly similar and highly correlated. To directly test the multiplex-GAM approach, we collected a new batch of 249 multiplex-GAM sequencing libraries, each containing 3 nuclear profiles on average from an independent biological replicate of mouse embryonic stem cells. We found that the quality of the 249 3NP dataset was at least as good as the 481 1NP dataset. To investigate genome-wide difference between GAM and Hi-C, we developed a method for directly comparing metrics derived from the two methods. For this analysis, we considered the context between loci with distance smaller than 4 megabits, given that the fidelity of Hi-C decreases at a larger genomic distance. Given that GAM and Hi-C data have very different numerical distributions, we first applied a distance-based z-score transformation to both datasets to address the distance decay. We then subtracted the two normalized metrics and extracted the most divergent context. That is, this for which the difference between the two metrics were greater than the 5% of extreme defined by a fitted normal distribution. We refer to this most differential context as GAM specific or Hi-C specific. To assess whether the context differential detected by Hi-C have important biological roles, we created a dataset of features including heterochromatin markers, transcription factors, RNA polymerase P2, and transcription-related histomarkers. We then counted the number of contacts in each category between each possible pair of features, and looked for feature pairs overrepresented or underrepresented from GAM-specific or Hi-C-specific contexts. We found that most feature pairs are more frequently in the site of specific contexts than in the genomic background. Most of the feature pairs shown a stronger enrichment in GAM-specific context than in Hi-C-specific context. Only a small subset of feature pairs are more frequently in Hi-C-specific context. We performed a random forest method to find the most important feature pairs that discriminate GAM and Hi-C specific context. We found 10 features that has the highest discriminatory power that enriched in GAM, Among these 10 feature pairs, 6 of them involved known architecture factors such as CTCF interacting with active features like RNA polymerase, P300 enhancers, or OCT4. Interestingly, CTCF heterochromatin contacts were also enriched in GAM-specific contacts. By contrast, heterochromatin regions such as H3K9ME3 or H4K20ME3 were the only features most enriched in the site of high-C specific contacts. Overall, we found that the multiplex-GAM is cost-efficient and as good as the original GAM experiment. GAM and high-C can detect similar topological associated domains, and many strong contacts, including the large portion of CTCF-mediated loops, are also detected by most methods. The strongest contacts, especially found in GAM, often involved active regions, including many transcribed genes and superenhancers, while in high-C there are more inactive regions. The active regions are involved in extensive complex contacts that are currently underestimated in ligation-based approach.